I surely had fun preparing it. So the topic today will be biophysical understanding of sugar metabolism from a physical perspective, using a combination of empirical and theoretical approaches. So I really tried, and I will try to keep the subject as simple as possible, because it is my understanding that the audience is diverse, and going into too many technical details is maybe not advisable. And then I try to kind of find balance here, and to present this in a, say, combined popular and scientific way. So let's dive straight, well, not really straight into the topic, because first I'd like to say a few words just about myself, because I suppose that, well, at least some people don't know me. As a scientist, I started out as a mathematical modeler, along the way kind of turned into a keen student of human behavior. That progression was, it seems maybe a bit strange at first, but there was a natural progression from ecological modeling towards mathematical biology. And in mathematical biology, the evolution of human cooperation is a big topic. And my former postdoctoral advisor, Jo Ivasa, who is basically one of the more famous mathematical biologists in the world, and definitely number one in Japan. So that was a big topic in his lab. And then when I joined, I was more into ecological modeling, and then switched to mathematical biology and started kind of naturally working on human behavior as well. I do biological physics, and nowadays I would say that is my main field of study, as I will show shortly. And then if time permits, I do a little bit of network science and science education as well. And this slide is also kind of to indicate that I am present on Twitter. So if Twitter is your thing, please feel free to join me over there. So as I said, the main thing that I kind of do is, so it's not really the sugar metabolism or any specific type of metabolism, but more the approach that I take is more general, and it's kind of summarized in this title of a review paper that we published, what is now already 3 years ago, so Physics of Metabolic Organization. So basically, it's an approach where we look at organisms in a way that is illustrated essentially in this figure. So we look at organism as a kind of a set of compartments. Specifically, there is a reserve compartment. It's where organisms, so when organisms eat food, they assimilate energy. That energy goes first into this reserve compartment. From there, it is used for different metabolic processes, such as growth, which serves to build up structure being everything, all tissues that is necessary for the organism to stay alive and to perform daily functions. And then some of that energy from reserve is also dissipated. Dissipation, again, it's for maintenance purposes, for development, maturation purposes, and so on. So it's an approach. It's basically a bioenergetics approach to metabolism, which results in models like this. So just for the curious people out there, 3 dynamic variables representing the reserve that I just mentioned, the structure, and the level of maturation. They are connected through 4 energy flows, which again depend on these state variables, on these dynamical variables, so that we can actually solve something, get a system of equations that we can solve. And this gives us various interesting types of results. And this is just one example where we basically used, on the left side, we used data we kind of ran the model in the inverse direction. Normally, we would input food and then get growth, maturation, reproduction. So basically, the whole life cycle of an individual from an egg to an adult individual, and then the eggs of that individual. So it's a complete life cycle model. Normally, it runs by specifying environmental conditions and then running the model in the opposite. So from environmental conditions to growth and maturation and reproduction. Whereas here we actually started from growth data, from some sort of data from this data on captive fish, specifically bluefin tuna, is Japanese data. And they were observing some curious phenomena with this particular group of tunas, and they couldn't really understand what was going on. So we tried our bioenergetic approach to see if we could shed some light of what was going on there. And we used this growth data, basically ran the model in the opposite direction, estimated the feeding history. And using that estimated feeding history, then we could see, again, run the model against, now using that feeding history, the estimated one, run the model in the normal direction and see that the growth, there was basically these fish were calorically restricted. They were not receiving an optimal amount of food. We also found out here on the right bottom panel that what we predicted, the estimated feeding history that we got out of the model pretty much was matched really well the data that they had. But now we could actually understood that these fish were not receiving enough food. That's why they were growing differently from what the optimal growth, this dashed growth curve here in the right upper panel. I hope you can see my mouse arrow when I'm pointing at things. So we could understand that they were calorically restricted, and we could, for example, explain why it took these fish. So these were captive fish reared for specific purpose of producing, so getting them to reproduce and to produce small fish in captivity. So basically, the idea was to close life cycle in captivity. And okay, sorry, I see that some people cannot see the pointer, but I'm not really sure what I can do about it. Except maybe explain a little bit in more detail what I'm trying to say. But okay, it's not really material for the, it's just an example of what we can do with this particular approach. And the thing that we could explain is why it took their fish 7 years to sexually mature, to become adults, when normally it should take 5 years, even in the wild. And in really optimal aquaculture conditions, it can take them as little as 3 years. So obviously they were doing something wrong, and we could explain what they did wrong in this particular case. Okay, so if mouse pointer is not visible, then I'll just try to explain things in more detail and say where I'm looking at in order that you can better follow. Another thing, so as I mentioned briefly at the beginning, another thing that I do a lot these days beside this type of bioenergetic modeling is actually this. It started as models, evolutionary models so the models of the evolution of human cooperation, but then quickly kind of changed from models to experiments where we figured out, okay, we are physicists. I mean, running models is nice, but we physicists also like to do experiments, measure stuff, and try to actually see that, to see whether these models that we are making, which look very nice and are really lovely and display a lot of interesting dynamic phenomena, but does this have anything to do with real human behavior? So we started doing experiments called, so the whole set of this type of experiments called social dilemma experiments, where we essentially put people in a situation where they have to choose between common good and their selfish interest. And then we tweak that sort of situations. So the default situation is really bad for cooperation. So people normally go for the selfish option. But then we ask, okay, how can we tweak this situation? For example, introduce a little bit of recognition between players, or try to trick them with some sort of extra options that are really bad, but could still kind of affect their behavior and trick them to go for a more cooperative option. And an example of this, just an experiment that we have performed recently, would be kind of a climate change game. So where people are supposed to invest their wealth, their initial capital, into fighting or mitigating climate change. And we kind of divided people into three groups: poor, kind of middle wealth group, and then rich group. And we were looking how they behave. And the interesting outcome from this particular experiment was that essentially wealth inequality helps towards the solving. It's actually helpful for mitigating climate change. And we found that so both poor and rich groups invest more than their fair share. So they kind of participate actively. They do so for different reasons, but they are the kind of main engines behind a successful outcome. Rich people, in the experiment, rich people move and are active and invest more than their fair share, simply because they realize that they stand to lose the most. And in experiment that is, let's say, fairly obvious, so people figure it out, and then they become very active because they simply see that if they can help resolve the problem, then they'll take home a decent amount of money. They really do get real money. And then, but poor people really kind of feel the need to participate and contribute. So they are not, they definitely have kind of, it's not so calculated, it's not so rational, it's a human trait, and people just try to be in there. It's the middle, kind of these people who have, whose wealth is in the middle, who are kind of hedging one way or the other. Will the group succeed or not? They really calculate a lot, and they are the ones who kind of in the end underinvest and do not contribute to resolving the situation. In the real world, so to make this actually applicable in the real world, is we really need to find a way to get to those rich heads and make them realize that if climate change really happens, that they are the ones who stand to lose the most. When we do that, I guess they'll start moving, as do people in our experiment. But okay, that's what I do when I don't do network science. Today's talk is about network science, so I'll just go through a few preliminaries, and then we'll dive into the real topic of today's talk. So for those, I suppose that there are people who have never done anything with networks in the audience, so I'll try to explain, really start from scratch and try to explain first what a network is. So in kind of mathematical language, we say this is a pair of two sets. One set is a set of nodes or vertices, and the other is a set of edges or links. And the links basically connect two nodes. So this is what this mathematical statement here is saying. And then if we have, a network can be directed, so there can be a link from node X to node Y, but it does not necessarily mean that there is a link the other way around. If for all XYs, so all edges that connect nodes X, or all links that connect nodes, so for any two nodes, if we take any two nodes and there is a link connecting them, so then if there is a link in the other direction, then we say that the network is undirected. And we'll be working, so I basically in all my work so far have only used undirected networks. So today we are talking about such networks. And here in the right bottom corner is a very cute example of a network with 6 nodes, or with 6 vertices and 1, 2, 3, 4, 5, 6, 7 edges. So then when people talk about networks, especially some 10 years ago, it was very popular to talk about the structure of network. It's a kind of static perspective. And I guess because it comes, in large part, it comes from statistical physics. So when it's statistical physics, people like to find distributions of something, and with networks in particular, an important idea was the concept of node degree, which is just essentially for any node in the network that we select, we can just number the connections to other nodes, and the number of connections for a particular node is called node degree. And then people were interested in distributions of node degrees. And so the upper example, this is an example of a random network, or for maybe people who are involved in network science, this is an Erdős-Rényi network. And then on the right side, next to the network is its degree distribution. The network on the bottom is a scale-free network, so a very different type of network in which certain nodes have a lot of links, and then there are many, but those are few in number, but still they can have a huge number of connections. On the other hand, many nodes will have only a few connections. And when degree distribution of such networks is shown, it is often shown in a log-log plot like this one. And there, if it's a real scale-free network, then we should see a line like it is exemplified in this perfect case. So that was really popular maybe some 10, 15 years, 15, even 20 years ago. Later on, it became more popular to put dynamics on networks, to start considering dynamical processes on networks, or to make networks themselves dynamical. So I guess the simplest example is just to put a random walker on network and then examine different statistical properties, such as the first passage or time to return to original position, and stuff like that. Lately, it's very popular to do spreading processes on networks. Spreading processes because that's network epidemiology. So essentially, nowadays epidemiology is very popular for obvious reasons. So that is one really popular thing that people are doing a lot these days, combining. We do know from maybe 10 years ago a lot about network epidemiology. These days it's popular to combine that with behavior. So let's say there is one network layer in which the disease is spreading, and there would be on top of that another network layer where people, for example, spread rumors. And that rumors could affect. So people like to do, for example, things like vaccination is bad for your children, don't vaccinate your children. And then that sort of rumor spreads on one layer. And then depending on who the rumor reaches, then people respond to that in different ways. And that affects, of course, disease dynamics, which is then different than if there were no such, if that behavioral aspect was excluded. So that's kind of the state of the art of what people do at least in network epidemiology. And right here is an example that of basically it's an example of a dynamical network. So network itself is it's dynamical in the sense that nodes can be in different states. And depending on what's happening around them, nodes can change their states in time. And I'll explain how this particular network, so this donut network that you can see on the top, you can see that it's there is something called activity on the vertical axis, and then we can see on the horizontal axis, we can see that this is time. And we can also see that this network kind of starts in a very inactive state, then suddenly it can jump to an active state, this when it leads up, like here in the middle, becomes green. So nodes are doing something. Then it again spontaneously goes into an inactive state where nothing much is happening. So it jumps between these two states, and we can, with a little bit of control, we can make this more or less regular. So we can let the network do its thing and just jump between states as it wants, or we can impose a little bit of control, then it will do this more regularly. Why it is doing this is not really that important. The important idea is that network can be active, more or less active, and this changes through time. And I'll give, so the model that is used to generate these sort of signals is very simple, it's very generic, and I'll explain it using the example where we simply using more intuitive example than what later is happening with beta cells and sugar metabolism. So I'll just, the idea is almost the same, but just explanation here is a little more intuitive. So I'll go with this, and then we'll kind of transfer that into or adapt it to the situation with beta cells and with for sugar metabolism. So here in particular, so in this example, we say, okay, nodes, network nodes are neurons, and links between nodes or edges between nodes are synapses. As simple as that. So we are representing a biological system, in this particular case, a bunch of neurons in the brain, as a network in which, so each neuron, each cell is a node, and then neurons are connected, and those connections, they can communicate, and this ability of neurons to communicate with one another, we will represent in our network as links. So cells that can communicate, they are connected, there is a link between them, and cells that cannot communicate, then they don't have links between them. Then we say, okay, so neurons can be in two states, active and inactive, and any neuron can spontaneously jump into this from an inactive state to an active state, and when it does that probability P, or specifically this probability rate P, and then in a time interval delta T, it will jump with probability P delta T to this from inactive to active state, and it will remain in that state for some time. So these are very simple assumptions. There isn't much to it. Basically, the assumptions are very generic. I mean, I don't really have to talk about neurons. It could be pretty much anything that can change state from state 1 to state 2, and it can do that spontaneously, or as we have in the next slide. So neurons, when they activate, they can also kind of stimulate their neighbors to become active. So in the same way, it could be a more generic situation would be just a node is connected to another node, and when one node spontaneously on its own becomes active, then activity of that node can somehow affect another node, and that other node can also become active. So basically, there are two ways for nodes to become active. One is intrinsic, does node jumps from one state to another or on its own, and another way is extrinsic, so through mutual connections where one node affects the other. So with neurons, so when one neuron fires a signal, becomes active, that will spread through synapses, and then if the signal is strong enough, so there is with certain probability, the next neuron or several neurons that are down the line from our initial neuron will maybe become active, maybe not, but they become active because the first one stimulated them. And in this particular model, we were saying, okay, if there is just one neighbor that is firing, then there is one probability for neighbors for connected nodes to become active. And if one node has two or more active neighbors, then there is even higher probability that the node will become active. So basically, activity of a node is determined by its own internal probability to activate and by what neighbors are doing. The more neighbors, if there are more active neighbors, then the node itself is more likely to become active. So when we set up, and these are just 4 very, very simple rules that we use. Basically, it's almost like 3 rules. We just say, okay, nodes can be active. Nodes represent something, some biological system. They can be active or inactive, and they can activate in 2 ways. And that's what we are saying. Then we can look at the fraction of active nodes, play a little bit with probabilities and equations, and after a little bit of algebra, we came up with this equation 1 here. And what is important about it, that we have A, so the fraction of active nodes on left and right side, and in fact, the equation itself is nonlinear. Nonlinearity means that in some cases we will have multiple solutions. So for some set of parameters, we will have multiple solutions, and then we can get beautiful situations like here, where in parameter space. So if we look at different probabilities of activation, where for example, on the left panel, on the horizontal axis is just the probability of spontaneous activation, so intrinsic activation. Then there on the y-axis is the probability of activation if there is a one active neighbor, and then we fix the third parameter, so the probability of activation if there are more than if there's more than one active neighbors. And when we look in this phase plane, what is happening, we can see that there are essentially 2 distinct phases. Red color shows the active state, so no matter, or active phase. So no matter how we start the system, if the system is initially completely inactive, or if we start with a little bit of activity, or no matter how much activity we start, the system will end up in equilibrium of high activity, so in the high activity phase. The blue part of the phase space is the opposite. So no matter where we start, the system will just die out, kind of become almost completely inactive. Just some spontaneous firing will be in the system, but for the most part, it will be inactive. And then the white part is bistability zone. So we can have, at the same time, we can have both things. And that is depicted in on the right side, where we now on the vertical axis. So we again we fix 2 parameters. We'll add the third one, so just one of the activation probability slide, and we look at the number of. So for a randomly chosen node, we look at probability that we look at a fraction of its neighbors that are active, and we can see that. At the same time, for the same set of parameters, we can have 2 states. We can at the same time have stable active equilibrium, the red one, and stable inactive equilibrium, the blue one. And in fact, if we set the parameter just right, so if we set parameter P2, this one on the horizontal axis, to about 0.4 near this bifurcation point, what will start happening is that spontaneous phase flipping. So the system, because the system is stochastic, it can fire spontaneously. There is some spontaneous activity, and these things are never probabilities are what they are, parameters are what they are, but they get realized in a different way from time step to time step. Will sometimes land above this unstable green dotted curve. Sometimes we'll land below. When we land below, the system will simply go to the down state, to the inactive state. And then after a while, when stochasticity moves it above this green curve that represents unstable equilibrium, the system will then jump up spontaneously to the stable equilibrium. The important thing is that all this happens, for example, these jumps between phases are essentially a collective phenomenon. It happens simply because nodes can communicate, somehow exchange information, can communicate, or simply stimulate one another. So in that sense, it's a collective phenomenon, and this will be interesting to us when it comes to sugar metabolism, which is precisely so. Now we are finally, sorry for this long introduction, but now we are finally getting there. This is research that has just recently been published. I think it got the paper got an issue number over the past 2-3 days. The more technical titles, so for presentation, I decided to go with a more with a friendlier title. The more technical title is the one that you can see here, and it's a work that has involved quite a number of people. So some of you may know Professor Podobnik, who is, I guess, does not need much introduction in Croatia. Then there is a team from Slovenia of mostly physiologists who did the splendid empirical work and really helped a lot with interpretation. So these are physiologists that actually we physicists or mathematical modelers can talk to. And then a bunch of people, one colleague from Boston, then my boss here in Tokyo, and myself included. So it was quite a nice international collaboration. And if I had to summarize the idea behind this research in one slide, so this would be it. So we start with the biological system. We find a model representation in the sense that I just explained for neurons. So here we have on the left side these purple yellow thingies. These are blobs. These are essentially beta cells, a type of pancreatic cells that are involved in glucose metabolism and glucose homeostasis. I'll say just a little bit more about them in a moment, but then we go from this biological system to model representation, where we say, okay, cells are connected somehow. They can exchange information somehow. We will represent this as a network where each cell will be a node in this network, and then if two cells can communicate with one another, then there is a link between them. Once we set up a model, we look at the explanatory power of that model. So can it explain phenomena that we are observing and how well it can do that? And then if our model is, so the explanatory shows a great explanatory power, then we will claim victory. We will proclaim that, okay, this research has been a success and we have done it. So a few introductory remarks just to kind of put things into biological context. So these pancreatic beta cells participate in glucose homeostasis by producing, storing, and releasing the hormone insulin. And what is interesting about these cells is they maintain intracellular insulin reserve that is like 2 to 3 orders of magnitude above the lethal dose. So insulin is very powerful, and if not administered properly, very lethal. And for some reason, our beta cells have huge storage. They maintain huge amounts of insulin. And because it's such a potent, strong bioactive substance, well, handling it really demands precision and great care. And what we really want to know is how do beta cells do it? For us, in this approach, because it's a network science approach, what is really crucial is that beta cells are coupled via something called gap junctions. So it's essentially connections through which they can exchange electrical currents. And cells use these gap junctions to form what is biologically termed a functional syncytium, or essentially you can think about it as a cell collective. And our hypothesis for this particular research is that due to mutual communication, the nutrient sensing and insulin release emerge as collective processes. So just as with the brain thing, where previous example and explanations, where I explained the kind of basic modeling idea behind all this, where we had collective phenomenon of these phase transitions between spontaneous phase transitions between active and inactive phase. Here we are saying, okay, that insulin release and nutrient sensing are collective processes, and it's crucial for beta cells to actually communicate with another to function properly. So let's see just a little bit about the empirical side of this research. The team from Slovenia that I mentioned, so they will measure raw signals like this, where they are looking at so these currents that go through gap junctions. These are essentially ion exchanges, calcium in particular, and they will measure this sort of signals with negative trend and some periodic component, and then there will be some very fast oscillations involved. So when we on the bottom panel B, when we detrend the signal, that's how it looks. So there is some periodicity here, maybe characteristic time period is about 5-10 minutes. Then and on top of that there are these fast oscillations. So when we take a low pass filter, so we see these oscillations more clearly, and with high pass filter we actually or was this supposed to be the other way around? Anyway, our interest was to isolate these fast oscillations on the bottom that you can see in panel B, and we focused on these slow periodic oscillations were studied more so far in the literature, much more than fast oscillations. So we decided to go for this, and especially recent evidence is kind of showing that fast oscillations, these fast signals, may actually be more important for glucose homeostasis. So then we took. Here is a kind of detail of the signal over a time scale of 20 minutes, and we try to kind of compare these measured signals with model results. And model is essentially the network, pretty much the same as the one I explained for neurons, with the difference that why neurons are more intuitive than beta cells is that neurons activate one another. And that is kind of natural way to think about it is when one neuron starts firing, it will excite its neighbors, and then those neighbors will start firing, and there is a kind of a cascade or kind of chain reaction effect in the brain. It's controlled. It's not uncontrolled firing, but it's kind of signal gets multiplied as it moves through the brain. With beta cells, we have the opposite thing. They are kind of trying to control one another. And when there is too much sugar, too much glucose in the blood, then they release that control and they start. So basically, they communicate to their natural state would be to be active, but then they communicate with one another and keep each other at kind of at bay. And only when there is too much glucose, when there is a lot of glucose, then they release that control and they start firing. Or not really firing, in this case, start releasing insulin. So the point is that we want activity of our network to match these measured empirical signals. And we do that, we fit, so we use a fitting procedure to find parameter values for modeled signals that then for model outputs to really match empirical signals. Okay, and we can do rather well. So we get some statistical properties matched. We find parameter values that give us similar variance, similar skewness, similar kurtosis. And then, so this is called, for people familiar with it, it's called method of moments. And then we say, okay, so are we really, do we really have model that is explaining something that has some explanatory power that really is in statistical sense, that model signals and empirical signals actually match. Again, it's in statistical sense. So we can look at stuff like this, for example, the distribution of fluctuations that I have just, these fast fluctuations that I just showed to you. And we can see that indeed what we get, data that we have and the model that we get are statistically indistinguishable. They produce fluctuations that follow Levy alpha stable distribution, and that sort, so which is very different. As you can see, it's very different from normal distribution here shown with this dotted curve. And generally, this kind of Levy stable distribution, when it pops out, it's a kind of signature of complex system, connectedness, correlations in the system. So it is kind of pointing us in the right direction that we really do have some collective phenomena going on here. Then we looked at other things. For example, we binarized both empirical and signals that we get from the model. We looked at this delta t shown in the upper panel. It's called non-spiking interval. So this is essentially a time that is kind of activity of the network relatively low, or activity of beta cells, real beta cells, relatively low. And then when we look at distribution of these non-spiking intervals, so what we have from the model and what we have empirically really matches nicely. We could also very naturally, using this sort of model, so attaching a coupling strength. So because we have nodes and links between them, or edges between them, we could attach coupling strength to those edges. And then depending on depending on their activity, this strength could go up if nodes, so if cells are very active. This is actually what real measurements show in the bottom panel. So when two cells are very actively communicating, then their coupling kind of, which is measured by conductance, kind of goes up. Or when they are communicating less, then it goes down compared to the average. But for normal physiological glucose concentrations, so physiological in the sense that these are high glucose concentrations because they activate beta cells, but not enormously high glucose concentration. So as long as we are within physiological limits, statistically this conductance is conserved, and we could easily get something like that out of the model and explain why we should actually have that sort of conservation law. that again arises statistically. So it's not, this thing is not encoded in any particular cell, and it's not like it's not encoded in nodes. They are not programmed to preserve coupling cell. It just arises. It's just a statistical property that arises from some simpler rules of the system. If we want to look a little bit more under the hood, so just again at the model and some of the model properties. So in a sort of schematic way, this plot on the left, panel A, yellow and purple. So essentially, we have a forcing variable. In this case, with beta cells, this would be glucose concentration. Then we have coupling parameters. So essentially, how strongly two cells are connected on average. So what is the, or in more precisely would be a probability that the control, so I said beta cells, unlike neurons, beta cells kind of keep each other in check. And this coupling parameter gives probability that the control by neighbors will be released and the cell will be left to be as active as it wants to be active given the glucose concentration. So in this sort of parameter space, let's call it parameter space, we can again have very similar to the model with neurons. So we have a zone of low activity up. So unlike neurons, it's reversed, and it's reversed precisely because beta cells keep each other in check, whereas neurons motivate one another or prompt one another to become more active. So low activity is up, high activity is down. So it's forcing variable. In essence, glucose concentration. So if you look at the vertical axis, so high glucose concentration would be down, and low glucose concentration would be up. So as glucose concentration increases, we are approaching towards high activity phase. But it's kind of important, as we will see in a moment, it's kind of important to be near this critical point where the bistable zone appears. And this is just schematic representation of what we expect from this type of model based on its simple assumptions, essentially the same assumptions that I explained at the beginning. And then when we do numerical simulations, we really see that we get this sort of diagram that indeed shows a zone of high activity, a zone of phase of low activity, and then bistable phase. And this bistability really opens for when cells are very strongly coupled. We actually don't think that beta cells are so strongly coupled. They should be on the left side of the critical point, but still, from what our results suggest, they reside very near the critical point. And this is exactly what enables them to respond to glucose collectively. So it is shown in this particular plot. So you can see that low glucose is on the right, high glucose levels are essentially on the left, and then we are increasing glucose concentration. At the beginning, system is responding linearly, becoming more and more active, but just little by little, until it reaches a certain critical threshold glucose concentration. So in mice, for example, that is a 7 millimolar concentration of glucose. It's thereabout is a critical point. And we, in our experiment, we start with 6 millimolar solutions and increase during the experiment concentration to 8 millimolars, then decrease back to 6. And we see exactly this sort of sudden change in activity of cells. So the difference between these two, here are results for two different situations. One is with circles are for weaker coupling, slightly weaker coupling, and pluses are for stronger coupling. So with really strong coupling, we actually have the system inside this bistable zone, and then we can see hysteresis curves like this. But we believe that actually more physiological is what happens, what circles are showing here. So it's just that cells are not responding, not much, and then when glucose concentration reaches critical level, then the activity explodes, and when the concentration is returned back, they again become inactive.
Excuse me, Marko, may I
Yes.
just with a question? What I would like to understand are some links to animal or human body in different states. So does these two cases, do they represent healthy person versus...
Circles represent healthy situations. Pluses, I don't think that we observe actually pluses in biological system. We might, because it's also real biological system parameters are not really fixed, and coupling will essentially go. So I was showing the graph that was explaining that coupling on average is conserved quantity. Now we see evidence that we really go for non-physiological concentrations, so much higher concentration of glucose. This conservation law breaks down, and coupling is essentially gets stronger between cells. So it is possible that at some non-physiological concentrations, this sort of behavior, pluses, could be observed, but essentially a healthy biological system is circles.
Mm-hmm. And what about a person with sugar disease? Would it be then closer to the one with crosses in comparison?
That really depends. So in type 1 diabetes, it's actually what happens is that beta cells are kind of losing their function. And that would make the system more linear, actually. It would push it. So if we did not have, the point here is that...
I would decrease, not increase. Is this correct?
Yeah, so exactly. So if coupling went down in this particular model, so we would see here more and more linear responses. Pluses are for really high coupling. R is what we get when we fit model outputs to empirical signals. With lower R values, we would get a more linear response of the system. So we would essentially be moving away from the critical point here on this slide, but let's say on panel A, but we will be going left, not towards the bistable zone, but away from the bistable zone. And essentially, so I mean, this sudden response to threshold glucose concentration is precisely what I mean when I say that beta cells sense nutrients and release insulin, and they as a collective, it's a collective property. They do it as a collective. If each cell, if cells are looked in isolation, their response is actually linear. So this type of response is not more or less linear. I mean, it is. This type of response is not really encoded in individual cells. Only when we have really functioning collective of beta cells and they communicate with one another, we get this sort of response where they keep each other in check, and then they release this control, and suddenly the activity picks up at threshold glucose concentration. And this is what I mean by collective property.
Thank you very much. Yes.
Yeah, of course. If there are other questions, I'd be happy to answer if I can, of course. So other things that we see, for example, as glucose concentration goes up, when we reach threshold concentration, so we see that so this is standard deviation of activity. We see that this kind of explodes. So we see this sudden jump in activity, the response of the system, essentially threshold response, nonlinear response of the system. Another big thing that we try to look at were cross correlations. And what model says about cross correlations as opposed to what we get from the real system. So we can measure with nodes, we can look at the activity of individual nodes. With in measurements, they can they essentially get a plot of. They even measure distance in pixels. So essentially, they can look depending on the distance between 2 points in their data sets, distance in pixels. They can say, okay. Assumingly that we are moving from one cell to, as the distance in pixels increases, we are moving from one cell to its neighbors and then further neighbors and further away neighbors. And then when we look at what we should expect, at least qualitatively from the model, we see that in the inactive phase, cross correlation. So these are we have three curves representing cross correlation between first neighbors, second neighbors, and third neighbors in the network. So definitely we see that first neighbors should be more correlated than second neighbors compared to third neighbors. But we should also see that as glucose concentration increases and reaches this threshold, at that point we should see that the correlations really jump up. So the correlation should be much stronger, considerably stronger in active phase than in inactive phase. And when we look at empirical signals, it is precisely what we get. This is some sort of exponential. How was it? What was it? I know in the end that decays with distance is more faster than exponential decay. Anyway, really what's really important here is that in active phase we really measure stronger cross correlation across larger distances. And now this is new stuff. So this is the stuff that has not been published as a part of the first paper, but we are now working on this, analyzing data, and we are actually preparing this second publication on the subject, and especially focused. This publication will be focused really only on correlation and correlation networks.
In particular, recording now because you said you were speaking about some new results. Just let me know when I should.
Okay, yeah. So at this, if you want to stop recording, I mean, it can be cut out later, or you can just stop recording now.